So today I am going to discuss another antibiotic group, aminoglycosides. And aminoglycosides is also called aminoglycosidic aminocyclitols. Under this group, there are many antibiotics came, and that antibiotics are gentamicin, natilmycin, tobramycin, amikacin, kanamycin, streptomycin, neomycin. So these are the antibiotics. They belongs to group aminoglycosides. So before going to start the topics, we are going to discuss first the salient features. So in salient feature, few points are very important, and what the points are. First, this group is polycations. Polycations means they possess the positive charge. Usually, this group antibiotics came in the form of sulfate salt. They have the combination with the beta lactams, and if we are going to combine with the beta lactams, as a result, we get the combination which is a synergistic combination. This group is acting against mainly gram-negative bacteria and against the gram-negative bacteria, and which should be aerobic, not against the anaerobes. This group is also acts against the tuberculosis. And one of the antibiotics that belongs to this group is paromomycin. Paromomycin is usually used for the therapy of intestinal amoebiasis. Usually, we have seen the drug that are going to inhibit the bacterial protein synthesis are bacteriostatic in nature, except aminoglycoside. Aminoglycoside is bactericidal in nature. However, the other antibiotics that inhibit the protein synthesis are bacteriostatic. In antibiotics that inhibit the protein synthesis of the bacteria either by inhibiting 30S ribosome and 50S ribosome. In groups, there are only two groups, aminoglycosides and tetracycline. Both the group are acting on 30S ribosome of the bacteria. Regarding absorption of this group antibiotic is concerned, they are usually very meager absorbed or not absorbed. Why this group is not absorbed? Because they are polycationic. That means they are having the positive charge. So usually charged compounds should not be absorbed from the gastrointestinal tract. So that is why this group should not be administered by oral route. Almost all the antibiotics that belongs to this group are excreted out through the kidney. So excretion is through renal. This group have one another drawback that have the serious side effect, and the serious side effect concerned with the two major organ. One is the ear and another is the kidney. So this group is going to produce both kidney toxicity, that is nephrotoxicity, and ototoxicity, that is the ear toxicity. So because the toxicity is concerned to eighth cranial nerve, so this is concerned to the eighth cranial nerve damage. The drug is alkaline in nature, so they are having the alkaline pH. So this is all about the salient feature this group have, and this is the nutshell or gist of all the things that are possessing those aminoglycoside groups. Now we have to go through the history and source. Regarding history is concerned, in this antibiotic aminoglycosides, the first drug which was discovered was streptomycin. And this streptomycin was the first antibiotic, and this having the antibacterial activity, particularly against the tuberculosis. So this is the drug used for the therapy of tuberculosis, and the was developed or discovered by the scientist Wackersman and co-workers. So this is about the discovery of the first antibiotics from this group. The next important point with this group is that the natural and aminoglycosides are derived from actinomycetes. If they derive from the streptomyces, we are going to put suffix mycin. If the antibiotic get derived from another actinomycetes, that is micromonospora, we put the suffix mycin, but the spelling is MICIN, not MYCIN. So this is the difference, and this is indicating the actinomycetes type. Now we have to go through the classification of this antibiotic groups, or you can say this group aminoglycoside on the basis of its isolation is divided into two groups: the natural aminoglycoside and semi-synthetic aminoglycoside. Natural aminoglycosides, as earlier I have mentioned, isolated from actinomycetes. So the drug that isolated from natural actinomycetes are streptomycin. So this one is isolated from Streptomyces griseus. Gentamicin, that one is isolated from another actinomycetes called Micromonospora purpurea. Then neomycin, this isolated from Streptomyces freudii. Kanamycin isolated from Kanamyceticus, easy to recall because Kana is coming from Kanamyceticus. So this Kanamycin has been isolated from Streptomyces Kanamyceticus. Tobramycin is also easy to recall because the species is starting with the T. So tobramycin has been isolated from the Streptomyces tenebrarius. And another antibiotic that are semi-synthetic in nature, other than this are semi-synthetic in nature, and the semi-synthetic aminoglycoside are amikacin, natilmycin. This amikacin has been isolated or not isolated is the derivative of kanamycin. So this one is semi-synthesized from the kanamycin, I mean amikacin. Another semi-synthetic aminoglycoside is natilmycin. This is the derivative of sisomycin. However, the sisomycin has been isolated from the uh, Micromonospora genus, and the antibiotic was sisomycin, and from this natilmycin was derived. Other than these aminoglycosides that I have already mentioned, others are arbicacin, isibamycin, sisomycin. So this is all about the source and history. Now we have to go through the other point, that is the chemistry. So if you go through the name is aminoglycoside. So amino is indicating these aminoglycoside are possessing two or more than two amino sugar, and that is going to connect with another uh, compound we call either hexose, this is also a sugar, and this hexose is also called aminocyclotol. So aminoglycoside are having two or more amino sugar group that is going to connect with another hexose or aminocyclotol by a bond. We are calling it the glycosidic. So name of this antibiotic group is aminoglycosides. This aminoglycoside, if we going to put all the name together, then you may call it aminoglycosidic aminocyclitose. So this is either we have to call aminoglycoside or aminoglycosidic aminocyclitose. So this is the name. If you are going to put only the hexose or that is also known as aminocyclotol, so the aminocyclotol or only hexose that is the amino belongs to this group is spectinomycin. So what is spectinomycin? Spectinomycin is aminocyclitol or hexose that don't have the amino sugar. So aminocyclotol example is spectinomycin. So this is about the chemistry of aminoglycosides. Next is the classification of the aminoglycoside. So aminoglycoside has been divided into three types on the basis of their activity against the gram-negative bacteria that are narrow spectrum, broad spectrum, and extended spectrum. So in narrow spectrum, only one antibiotic is there, that is a streptomycin, and from this, when we are going to put the hydroxy group, then the name is the dihydrostreptomycin. So under the narrow spectrum aminoglycoside, there are two aminoglycoside. One is a streptomycin and another is dihydrostreptomycin. Next is the broad spectrum aminoglycoside. Under the broad spectrum aminoglycoside, most important are neomycin, kanamycin, and parmomycin. Neomycin and kanamycin is important because these are in these two neomycin is we are that this neomycin is in use. So neomycin and kanamycin both belongs to broad spectrum aminoglycoside, and the last one under the classification of an aminoglycoside is extended spectrum aminoglycoside. In this, the one is the derivative of kanamycin that we are calling it the amikacin. Then another is the gentamicin. This is also too much used in the practice. So gentamicin, tobramycin, amikacin, these three aminoglycoside that is coming under the extended spectrum aminoglycoside are too much in use. Other than these extended spectrum are sisomycin or and natilmycin, and these all are, I mean, genta, siso, and natilmycin, these all are isolated from the actinomycetes micromonospora, and this amikacin is the derivative of kanamycin. So and this tobramycin is the isolated from tenebrarius. So this is all about the classification of aminoglycoside. Now we have to go through the other point, that is the mechanism of action, or also known as pharmacodynamics. So this is very, very important. So in this, first we have to discuss this, as I have cited or mentioned that this is the only antibiotic group, that is the aminoglycoside, which is a protein synthesis inhibitor and is bactericidal in nature. So this is very, very important. Not only that, this group is rapidly bactericidal. So this is very, very important. But the problem with this group is that this is only acting against the gram-negative aerobic bacteria, not anaerobic bacteria. So because this is the narrow spectrum, you can say this is the narrow spectrum type of activity, because this is only or mainly active against the gram negative aerobe. So this is not, this is the antibacterial activity is not the broad spectrum type of thing. So this is very, very important. Another property this group have, that we call it PAE. PAE stands for post antibiotic effect. So this property is only shown by three group of antibiotics. One is fluoroquinolones, another is the aminoglycoside. So these two groups are possessing this post-antibiotic effect. So first we have to define what is the meaning of post-antibiotic effect. So the name is post-antibiotic, means कि after the antibiotic. It means post-antibiotic effect means if the concentration of the aminoglycoside fall below its minimum inhibitory concentration, the drug is not usually going to act upon the bacteria. But this group, along with the fluoroquinolones group antibiotic, even though their concentration fall below its MIC or minimum inhibitory concentration, they possessing the antibacterial activity. So that is why this is very, very interesting as well as the important. Third most important point is that first we have to discuss before going to start how the drug is going to kill the bacteria. First we have to see how this aminoglycoside get entered inside the susceptible bacteria. This aminoglycoside is going to first penetrate the outer layer by passive diffusion. That passive diffusion is through the aqueous channel. That aqueous channel is formed by the porine protein. So through porine protein, the drug is going to comes in the periplasmic space of the membrane, periplasmic space of the membrane. That means the outer layer penetrate the drug by passive diffusion and it reach to the periplasmic space. So once it reach in the periplasmic space of the susceptible bacteria, it has to reach in the cytoplasmic. So for that, this has to pass out the cytoplasmic membrane. So the second step of the drug to pass out this cytoplasmic membrane to reach in the cytoplasm of the susceptible bacteria, they pass out by a mechanism where it required the energy as well as it required the particular pH. It also required the oxygen. So if oxygen is not there, the drug should not penetrate the cytoplasmic membrane and the drug should not be reached to the cytoplasm. So this the second phase is known as energy dependent phase 1. The name is clearly indicating the aminoglycoside pass out through this membrane by with the help of energy as well as it also required the oxygen. So with the help of energy as well as with the help of oxygen, the drug should reach in the cytoplasm by the cytoplasmic membrane with the help of energy, and this is called energy dependent phase 1. So here the important point is that if the bacteria is not aerobic, I means is the if the bacteria is anaerobic, then the drug should not be passed out. So the condition to pass the drug is the oxygen should be present there. After that, once the drug it reached to the cytoplasm of the susceptible bacteria, as we already mentioned, or I have already mentioned that amongst the protein synthesis inhibitor, there are only two antibiotic group. One is tetracyclines, another is the aminoglycoside. They are acting on the 30S ribosome of the bacteria. So this one is the 30S ribosome of the bacteria. This is the 50S ribosome of the bacteria. And because here is the more than one ribosome, so we are going to call it the polyribosome. So this is polysome or polyribosome. So this is 30S. This is 50S, and this mRNA is found in the with the 30S ribosome, or and this mRNA having the sequence of the gene. Three genes are responsible for a particular amino acid. So with this particular sequence, amino acid is going to attach at the A side, that is a acceptor side, and at Another site is the P site. The P site is possessing the nascent protein. So how this drug is going to act? Drug enter inside the cytoplasm of the susceptible bacteria and going to bind with the 30S ribosome of the bacteria. So here the drug is bind. And how this is going to kill the bacteria? So once this aminoglycoside is going to bind with the 30S ribosome, and therefore what will happen? This AUG, this is called ribosome messenger RNA, tRNA initiation complex. Because this is the initiation complex. Why we are calling a protein synthesis initiation complex? Because here is ribosome, mRNA, and tRNA is going to combine together and form the initiation of protein synthesis. So initiation is of the protein synthesis start with the gene sequence. We call it the AUG. So this complex is called initiation complex. In this initiation complex, we will found 30S ribosome, so ribosomal, then mRNA, so messenger RNA, and tRNA initiation complex. So once this aminoglycoside bind with the 30S ribosome, it block the initiative of protein synthesis. So it block the initiation of protein synthesis, first step. So here this is going to block the protein initiation of the protein synthesis. Even though some of the bacteria, they are proceeded in some forward step, other than this initiation starting phase, then how this aminoglycoside is going to act? This is going to going to terminate the protein synthesis. How this is going to terminate the protein synthesis? Because this is going to misread the protein. What is the meaning of misreading? mRNA it initiate or block the first is the initiation complex, and second is the misreading. It inhibit it produces the misreading. What it means misreading? That means a particular amino acid is not going to attach with at a particular place in protein synthesis. So what will happen? Because of the misreading, mRNA misreading, the particular amino acid is not going to attach at a particular place. So what will happen? The protein that gets synthesized, that is not correct protein sequencing. So because of the incorrect protein and not complete protein gets synthesized, because of the improper sequence of the amino acid, the protein synthesis get blocked and fully protein get not synthesized. So what will happen? The protein that gets synthesized, we called it the aberrant protein. Aberrant protein is also called the faulty protein. So once this faulty protein get formed, what will happen? This faulty protein get attached to the cytoplasmic membrane of the susceptible bacteria. So once this aberrant protein in the second phase get attached to the cytoplasmic membrane of the susceptible bacteria, it initiate again the increase the permeation. It increase the permeation. Therefore, what will happen? The porosity of the cytoplasmic membrane get increased, and therefore again it start the permeation of more and more amount of aminoglycoside. By another phase, we are going to call it energy dependent phase 2. So what is this energy dependent phase 2? In energy dependent phase 2, aberrant protein is going to attach with the cytoplasmic membrane, and therefore what will happen? The porosity of the cytoplasmic membrane get increased, and more amount of aminoglycoside get penetrated and come into cytoplasm through a phase we are calling it the energy dependent phase 2. So more amount of the drug reach in the cytoplasm, and therefore they inhibit the protein synthesis, increases the porosity of the cytoplasmic membrane of the bacteria, and therefore it will not only inhibit the protein synthesis, also it increases the porosity, so the permeation of the cell membrane get increased, and therefore this protein synthesis inhibitor aminoglycoside is able to kill the bacteria. So that is why this is possessing the cidal in nature. That I already mentioned. It caused the misreading of the mRNA. It increase, and therefore this aberrant protein get attached to the cytoplasmic membrane, and therefore it increase the increase the permeability, and because of the altered permeability or increase of permeability, it further stimulate aminoglycoside transport through the another energy dependent phase. We call it the phase 2, and that is why this way the bacteria is going to be killed. So this is all about the mechanism of action of this glycoside. If you don't recall any more much more about these how these things happen, you have to recall by 2 3 points. The antibiotic aminoglycoside bind to 30S ribosome of the bacteria, and it inhibit the initiation of protein synthesis. It initiated the synthesis of aberrant protein. It initiated the permeation of more aminoglycoside, and therefore increase the porosity and inhibit the protein synthesis, and finally kill the bacteria. Therefore this is called the lysis of the bacteria, and therefore called the bactericidal group. So this is all about the mechanism of action of this one, aminoglycoside. Now we have to go through the another point, is that this group antibacterial activity of aminoglycoside, as I have mentioned, for its transport it require oxygen. So that means this aminoglycoside is mainly or only acting on the aerobic bacteria, and which should be gram negative. So aminoglycoside is primarily acting against the aerobic gram negative bacillus or bacilli. Another important point is that this group is going to combine with the other group to make it the synergistic combination, and that is most one usable or most tested combination, and this combination is called synergistic combination. Synergistic combination is the combination of the two antimicrobial antibiotics in which their killing effect is more than 1 plus 1, means ki more than 2, and this is most tested one. So how we are going to combine this? Penicillin is going to combine with the streptomycin, so that the combination is coming is streptopenicillin. So in this, either we have to going to combine with the penicillin or vancomycin with the aminoglycoside streptomycin mainly, but other than streptomycin we are also combining with the gentamicin. This is one of the most usable aminoglycoside. Gentamicin is the another one. So, amikacin is also used, tobramycin is also used, but the gentamicin is the one of the most usable aminoglycoside. So this one is the bactericidal in nature, and in combination it broaden the activity, and it act against enterococci, streptococci and staphylococci. So this is all about the antibacterial activity. Now we have to switch on of the another point is about the resistance, how this group of bacteria is going to develop the resistance against the aminoglycoside. The similar to the other antibiotic, they inhibit the penetration of aminoglycoside or antibiotic. So if the drug is should not be penetrated and should not reach in the cytoplasm, how the drug is going to kill the bacteria? So resistant bacteria is going to inhibit the penetration of the aminoglycoside. Another point is that if the target is get changed, if the if the resistant bacteria is going to change its target. So what is the target of this aminoglycoside? Is the 30S ribosome. So what the resistant bacteria is going to do, or this is going to change the target, that means ki 30S ribosomal structural. So a structural modification in 30S ribosome occur, and therefore this aminoglycoside is not able to bind with the target because the structure is changed. So aminoglycoside is not going to found the place where they have to bind, and therefore the bacteria is going to develop the resistance. So the second point is this one, penetration inhibition. So structural changes of the target. Next is the some of the resistant bacteria is also inactivate the aminoglycoside. How this aminoglycoside and inactivation is done by the bacteria. Bacteria, resistant bacteria is going to produce the enzyme, and that enzyme is going to act on aminoglycoside and modify the structure of aminoglycoside or break it. So therefore, the aminoglycoside is not active. And how the enzymes are? These enzyme resistant bacteria produce the enzyme. They are called transferase enzyme. These transferase enzymes are usually plasmid mediated, and these transferases enzyme are phosphotransferases, acetyltransferases, adenyl transferases. And the main and most important way of the resistance of this one is the natural resistance. So as earlier I cited that this penetration of the aminoglycoside require oxygen. So anaerobic bacteria, they don't have the oxygen there to to transport the drug into cytoplasm. So therefore, the anaerobic bacteria is the natural resistance against the aminoglycoside. So this is all about the resistance, how the resistant bacteria is developing the resistance against the aminoglycoside. Now we have to go through the other point about the aminoglycoside, what these are. The most important, as I earlier cited, the aminoglycoside are cations, polycations. Cations means they are having the positive charge. So usually the drug that possessing the charge are not absorbed. So that is why when you are administering this aminoglycoside that are highly polar, polycations in nature, they are not going to be absorbed from the gastrointestinal tract. Therefore, they are poorly absorbed. So therefore, orally should all aminoglycoside are not administered by oral route except only one drug, that is called neomycin. So neomycin is the only one aminoglycoside among all aminoglycoside that is coming in the oral preparation, and that the combination is one preparation is coming for the poultry, we call it the neodox. neomycin along with the doxycycline. So neomycin is the only aminoglycoside is for the oral route or per os route. Other than that, the others are for the intramuscular or subcut route, or you can say the for the parenteral route. So this is about the absorption of drug that I have cited. The drug should not be absorbed through the oral route. And the another point about the distribution. Regarding distribution is concerned, the similar to this cationic in nature, the drug is also having less penetration because of the charge. So less penetration is there, that means less distribution is of the drug or aminoglycoside should occur. So this is also less penetrable and less distributable. So other than the important point with the aminoglycoside is that, as earlier I in the silent feature I have mentioned, this is nephrotoxic as well as the ototoxic. The question is arises why this is nephrotoxic and not ototoxic, because this aminoglycoside or these aminoglycoside is going to deposit in high concentration in renal cortex as well as in the endo and perilimb of the inner ear. So because of the high concentration deposition in these organ, I mean renal or kidney or inner ear, the organ toxicity occur. So if the concern to the kidney, we call it nephrotoxicity. If this is concerned to the this ear, we are going to call it the ototoxicity. So this is the and next is the absorption is there, then distribution, then excretion is concerned. They are primarily excreted out through the kidney by glomerular filtration. So this drug is also should be used for the therapy of UTI infection, urinary tract infection of aerobic type, because this is acting against the only aerobes. So why the for use for the urinary tract infection? Because the drug should be excreted out through the renal or the kidney. That means the drug should reach there at high concentration. So that is why this should be also used for the urinary tract infection. Regarding dose is concerned, the dose is concentration dependent. So usually we are administering this group at high concentration, because high concentration is going to produce more killing effect, and one because this is the toxic in nature, so we are administering it once daily. So the drug of this aminoglycoside group is administered at high concentration and once daily dosage, and should not be used for longer time interval because of the auto as well as the nephrotoxicity. So this is all about the pharmacokinetic property of this group. And now last heading is the untoward effect concerned to the aminoglycoside. This is very important, because this is the group which poses very much toxicity to the major organ kidney and ear. This toxicity may be the reversible or may be the irreversible, depending on the deposition there. If the concentration is high and deposited for the longer time interval in the vestibular and cochlear region of the internal ear, then if the deafness occur, then this become irreversible. If the kidney damage is the major, if the damage is more, then again this become the irreversible nephrotoxicity. So this, and along with that, they are also going to block the neuromuscular blockade. So why this ototoxicity occur? As earlier I have mentioned, this aminoglycoside is going to deposit high concentration in peri and endolymph of the inner ear, and because of the high concentration deposition and less excretion from this ear, then what will happen? This is going to degenerate the sensory hair cell in organ of corti. So because of the degeneration of this sensory hair cell in this internal ear or inner ear, they going to produce the deafness. So This is irreversible type of. And if you are going to administer this aminoglycoside with the other type of the nephrotoxic or ototoxic drug like the furosemide, ethacrynic acid, cisplatin, they are also toxic. uh, they are also particularly nephrotoxic. So therefore, we are not going to administer this aminoglycoside with the other nephrotoxic or ototoxic drug. The important another important point with this aminoglycoside is that amongst the all aminoglycoside, which one aminoglycoside is the highest toxic one? And the example is neomycin. Neomycin is the highly nephrotoxic because they are going to deposit in a high concentration and not easily excreted out from the kidney once it get deposited. Therefore, they are highly nephrotoxic in nature. So they are going to deposit in proximal tubular cell. So therefore, it is going to produce the renal toxicity. So that I have mentioned that neomycin amongst the all aminoglycoside are highly nephrotoxic because they accumulated there in high concentration. So therefore they are highly nephrotoxic in nature. And that is one another reason the neomycin is mainly or only used for oral preparation because they are not going to absorb more in the systemic circulation. This streptomycin is amongst the all aminoglycoside is less nephrotoxic. And this is the only one antibiotics amongst the aminoglycoside which is possessing the antibacterial activity against the mycobacterium. So this is also used for the therapy of tuberculosis. Other than other toxic effect is that because this aminoglycoside is cation in nature, so this cation in nature means ki have the positive charge, and as you know, neurotransmitter acetylcholine at the nerve end require calcium to release the acetylcholine, and your aminoglycoside is also having the positive charge. Calcium is also having the positive charge. So because of this positive charge of aminoglycoside and positive charge of this calcium, this calcium should not reach in the nerve end, therefore acetylcholine should not be released. So aminoglycoside also going to inhibit the release of neurotransmitter acetylcholine, and therefore it produces neuromuscular blockade. And if the patient like myasthenia gravis, so myasthenia gravis are the patient, their muscles are weak, and to maintain its tonicity, the acetylcholine must be there in proper concentration. So when you are giving in these patient the aminoglycoside, then what will happen? Acetylcholine concentration is low, therefore this is going to create the problem. So in myasthenia gravis patient, we are going to avoid the aminoglycoside treatment. So this is all about aminoglycoside.